Myself, Dr. R. Sanap, Head and Associate Professor, Department of Botany, Rao Bahadur Narendra Boroke College, Rampur. Today I am going to deliver the lecture on the study of Pinus. It is the part of syllabus of T.Y.B.Sc Botany, paper 4th, that is BO 334, and title of the paper is the Spermatophyta and the Paleobotany. So in that there is a part like the Angiosperms and the Gymnosperms. So in this Gymnosperms, today I am going to deliver the lecture on the study of Pinus, in that the anatomical structure of the Pinus stem. So if we see the TS or the transverse section of the Pinus stem, then it shows the presence of the different types of tissue layers. So out of that, the outermost layer is the epidermis. So this epidermis, it is the outermost layer of the stem, and this epidermis, it is single layered. So this is the epidermis. So this epidermis, it is single layer. Cells are barrel shaped and compactly arranged without intercellular spaces. So this epidermis, it is covered by a thick layer of cuticle. So it shows the thick layer of cuticle. So this cuticle, it is nothing but a cutin substance. It gets deposited on the outer wall of this epidermis. So thus this is the cuticle. So this, on the stem of the Pinus, there are presence of the scale leaves, and so if we take the transverse section of the stem, then it shows the presence of the marking of the lip scars. So these are the markings of the lip scars of the scale leaves. Then this epidermis, it is followed by the presence of the cortex, and this cortex, it is multicellular. If we see the cells of this cortex, these are circular in outline. So these cells, these are loosely arranged, and so they show the presence of the intercellular gaps or intercellular spaces. And this epidermis, this cortex, it is multi-layered, and this region, it shows the presence of the there is a presence of the region canals. So there are many region canals are present in the cortical region, and this region canal, they secretes the substance, that is the region substance. It gets secreted by this region canals or the region ducts. So this is the cortex. Cortex, it is made up of the parenchymatous cells. These cells, these are thin-walled. These are loosely arranged without intercellular spaces. So we can observe many intercellular spaces in the cortical region. So this is the cortex, and it is multi-layered. So this is the cortex, and this cortex, it shows the presence of the region ducts. This region ducts, this secretes the region substance, and these cells, many cells of this cortex, they also show the presence of the tannin cells. So tannin cells are also present in the cortical region. So these are the tannin cells. So here, these are the intercellular gaps. So So these are intercellular gaps, or maybe called as the intercellular spaces, and that's why these cells, they becomes the loosely arranged. So this is the multi-layered cortex, and inner side to this there is a presence of the primary xylem. So here there is a presence of the primary xylem. So the cells of the primary xylem, so as the name indicates, xylem, the cells are dead and these are lignified. Dead and lignified cells are present in this xylem. So due to the, actually if you see this tiers of stem, so in the pinus, if you see the vascular bundles, so these vascular bundles, these are conjoint, they are collateral, and they are open type. Conjoint, collateral, and open type of vascular bundles are present. And when this secondary growth starts, so at that time the cambium, it produces the secondary xylem and the secondary phloem. So here the presence of this region, so it shows the presence of the primary phloem. So this is the primary phloem. Inner side to this there is the presence of the secondary phloem. So due to the formation of the secondary growth towards the outer side of this cambium, so here this secondary xylem, secondary phloem is formed, and due to this, here the primary phloem, it get pushed towards the outer side. So thus here, this is the secondary xylem, secondary phloem, and due to the formation of the secondary phloem, this primary phloem, it gets pushed towards the periphery, and here there is the presence of the cambium. So this cambium, it is single layered. So this cambium, it is the meristematic tissue. So that is the meristematic. It cut off the cells towards the outer side and also towards the inner side. So tissue which is developed towards the outer side, it is called as the secondary phloem, and tissue which is developed towards the inner side, it is known as the secondary xylem. So thus here later on this cambium, it also produces the tissue towards the inner side, and this tissue which is formed towards the inner side, so it shows the presence of the secondary xylem and also the secondary medullary rays. So that's here, this is the secondary xylem. Cells of the xylem, they are dead, they are thick walled, they are lignified. So as this tissue, it is the dead tissue, the cells, these are dead, that is cells, they are dead, and that's why it is called as the dead tissue. Cells, they become thickened due to the deposition of the lignin substance, and that's why it is, when if we stain it with the saponin, then it becomes the red colored. So that's here, this is the secondary xylem. Cells are polygonal in shape, compactly arranged without intercellular gaps or intercellular spaces. So that's here, this is the secondary xylem. So it does not show the presence of the intercellular gaps or intercellular spaces, as the cells are compactly arranged, polygonal in shape without intercellular spaces. So this is the secondary xylem, and it shows the presence of this medullary rays. These medullary rays cells are elongated, attached one above another. So these are the medullary rays. So due to the formation of the secondary medullary rays, here the primary rays, they get pushed towards the inner side, and here towards the central region, here due to the formation of this secondary xylem, so here this primary xylem, it gets pushed towards the central region, and it shows the presence of the metaxylem and also the protoxylem. So here this both this protoxylem and metaxylem, they are thick-walled cells. So see, this is the metaxylem, this is the protoxylem. Actually here this is the primary xylem. So due to the formation of this secondary xylem, the primary xylem, it gets pushed towards the center, and at the center there is a presence of the pith. So this pith, it is parenchymatous. Central portion of the stem, it is occupied by the presence of the parenchymatous pith. So thus here it is multi-layered pith is present. Cells of the pith, they are parenchymatous, they are compactly arranged, they are loosely arranged, and they show the presence of intercellular gaps. Beside this, it also shows the presence of the tannin cells. So here these are the tannin cells. So thus here this is about the internal structure of the stem of the pinus. So if we take the transverse section, the outline of the section, it is circular in outline, and many times it shows the presence of here the leaf scars. So when the plant, it becomes mature, then later on inner side to this epidermis, there is a formation of the periderm. Actually this periderm, it is not found in the younger stem, but as the stem it becomes mature, then the periderm is formed. This periderm, it shows the presence of the three types of tissue layer, that is phloem, phelogen and pheloderm. So this phelogen, it produces the tissue toward the outer side, it is called as the phloem. It produces the tissue towards the inner side, it is called as the pheloderm, and this phelogen, it acts as a meristematic tissue, and thus these three layers togetherly later on it form the periderm. And due to the development of the more secondary growth toward the inner side, pressure is exerted on the epidermis, and so later on the epidermis gets stretched more and more, and ultimately after sometimes it ruptures or breaks. And to protect the inner tissue, in the cortical region, just outer side towards the outer region, this some cells of the cortex, they become meristematic and acts as the cork cambium, or may be called as the phelogen, and thus it produces the tissue towards the outer side and also inner side, forming the phloem and the pheloderm respectively. And thus phloem, phelogen, pheloderm together it is called as the periderm, and thus function of epidermis later on it get performed by this periderm, that is the protection. So thus here this is about the different types of the tissue layers which are observed in the internal structure of the stem of the pinus, having the cuticle, epidermis, periderm, multi-layered cortex, in which there are presence of many resin ducts which secrete the resin, and some cells, they show the presence of the tannins, and cells are loosely arranged parenchymatous, show the presence of the primary phloem, secondary phloem, cambium, and inner side to it there is a presence of the secondary xylem having the medullary rays, primary xylem get pushed towards the central region, and here at the center there is a presence of the parenchymatous pith. So thus here this is about the internal structure of the stem of Pinus having the different types of tissue layers. So this is about the TS of Pinus stem. Now here I am going to conclude this lecture. Okay, thank you very much.